Hi, my name is Ryan Stephens, and I'm a postdoc here at University of New Hampshire, and I study small mammals, and especially their associations with mycorrhizal fungi.
Okay, so I see you have two skins here of flying squirrels, and I know it can be really hard to identify the differences between both of them unless you have them like up close in your hand and everything. So how would you go about identifying the differences of these two flying squirrels?
Yeah, that's a great question, and you're totally right. If you see them out at your bird feeder or something, it can be really hard to tell. But when you have them in hand, it can be a little bit easier. So this is a southern flying squirrel, and this is a northern flying squirrel. And you can see right off the bat, one's bigger. So the northern flying squirrel tends to be quite a bit bigger than the southerns. They're about 80 grams or larger, whereas the southerns, I think somewhere around 70, 75 grams or less. So there's a bit of a size difference there. You can also see some differences in the pelage or the coat. So the southern flying squirrel tends to be a little more gray-brown, whereas the northern tends to be a little more cinnamon or kind of a red-brown color, so that their fur is a little different. There's also, if you take a look at their tails, the northern tends to have this darker spot on the end compared to the southern that doesn't. But pelage or the fur color can differ a lot by area, so it's not always the most diagnostic. I would say the best way to identify them if you have them in hand is to look at the belly fur. So the southern, if you take a look, and the belly, the hair is white to the base of the skin, whereas the northern, if you take a look, it's gray at the base. And so that's probably the best diagnostic characteristic to use to distinguish them.
Mm-hmm. Okay, so how would you go about making skins like these for flying squirrels?
Oh, that's a great question. So to make a skin, we basically make a short incision in the belly and basically invert the skin out, and we remove all the flesh and fat and things like that. Sew up the mouth, we make a cotton body, and basically roll it back on and put some wires in the legs and sew it up and let it dry. So it's just skin right on cotton. So kind of a simple, not quite taxidermy, but a nice museum quality taxidermy.
Yeah.
Museum specimen.
You're
We
good.
also collect tissue samples, so when these go to a museum, if someone wants to do genetic studies, they can do that, and then we clean out the skeleton as well, in case someone wants to look at the skull or our postcranial parts.
Mm-hmm. Okay, and are you able to pick up any kind of information about how they live their lifestyle?
Yeah, that's a great question. So there's a number of analyses we can do. For example, we can take hair samples and we can use isotopic analyses to get at their diet. We can also look at reproductive conditions. So when we cut it open on females, we can look at the uterus to see if there's any scars or embryos to get an idea of how many pups they might have had. And for males, we can look at the testes size, which gives an indication of reproductive status as well.
Okay, and then last question. From your experience, do you feel there is satisfactory data on flying squirrels in museums or anything?
Great question. I guess I'm not too sure, but I think that especially lately, there's been less preparation of museum specimens that are going into museums, and I think that could be a problem into the future if we want to look at range distributions or changes genetically or other things. So I think there's probably fewer specimens going in now than there used to be, and we're probably missing an important record in time.
Yeah.
So, Ryan, can you tell me a little bit about your job, like what you do and how this relates, all these props?
Yeah, that's a great question. So one project I'm working on right now is looking at mammals, small mammals, and carnivores as dispersers of mycorrhizal fungi. So mycorrhizal fungi are really important in forests. They help trees grow, and they're really important for establishment. And mammals, especially small mammals, are important for digging up truffles and eating mushrooms and then dispersing their spores. But that dispersal happens at relatively short distances across the landscape. So one thing that I'm looking at now is the role of carnivores as secondary dispersers. So things like foxes and coyotes and wolves eating small mammals and then dispersing those spores even farther. So some of these specimens were collected as part of that project.
Okay, thank you. What's that sample?
Which one?
The slide.
The
Can you
slide.
talk a little about
Yeah.
the slide?
So when I put these specimens up as museum specimens, I collected some of the scat or the gut contents.
Okay.
And that's where I can look for truffle spores. So I go through a filtering process and then spread that, basically a spore isolate on a slide. And then I look under a microscope and count the number and types of mycorrhizal spores that I see. And then we'll also do some genetic analyses to kind of verify those taxa.
Okay, awesome, thank you. So what inspired you to start getting into this field, like ecology and all of that?
Wow, yeah. So it probably started from a young age. My parents really encouraged me to go outside and explore, and I got really interested in being outside, and I didn't really realize there was a career in that until I got into college. And kind of one thing led to another, and I got really interested in ecology and biology, and here I am now.
Wow. So why did you decide to study rodents, small mammals, and specifically this fungi disbursement?
Yeah, I got really interested in small mammals in my probably junior year in college, or actually sophomore year, and I took a class where we actually went out and live trapped small mammals, and I just thought it was the coolest thing. And then ended up studying small mammals for my master's research out in Wisconsin. And I came out here, and my advisor and I were really interested in diets of small mammals and wondering kind of what they're eating. And that led to kind of looking at their role in eating fungi and dispersing spores. And and I've been really interested in that ever since.
Okay, awesome. And how long have you been doing this? Have you been working and conducting these studies and researching these mammals?
Yeah, so probably for over 10 years now. I started my I started my undergrad in 2005, and so yeah, a while, longer than I care to admit, maybe.
Okay, wow, that's very impressive. So why is it so important for people to learn about ecology or research it on their own?
I think ecology is really important because it's kind of the basis of our functioning ecosystems, and ecosystems provide a lot of important things for us, especially forests, timber, and other resources, and a place to get out and see nature. And so I think understanding the processes that go into keeping those ecosystems healthy is really important.
I agree. So you mentioned on your website that you really like mentoring students, and you say it's one of the most rewarding experiences for you. So why did you start getting into that, and why do you think it's important for established scientists to mentor young adults into the field?
Yeah, so for me, I got started because researchers or faculty members showed interest in me and helping me kind of follow my interests. And so I guess I'm kind of returning that. And I think helping show students what's available and getting them exposed to research is really important and kind of sets them on a path forward.
So earlier you mentioned how you study how small rodents impact the mycorrhizal fungi disbursement. And one aspect of our study is how forest fragmentation impacts the population of flying squirrels. How would you say that would impact the mycorrhizal fungi disbursement?
Yeah, that's a great question. And so there is some evidence based on molecular data looking at soils, so these mycorrhizal fungi live in the soils, and stands that are cut and that are isolated from kind of intact forest have very different fungal communities than those that are right next to intact kind of older growth forest. And so and some of the first fungi that are coming in to stands that have been recently harvested and that are close to older growth forests are truffles and things that are dispersed by mammals. So I imagine that the more fragmented those landscapes get, the less dispersal you have by kind of the small mammals, those type of critters, and probably you have more communities of fungi that are wind dispersed.
Okay.
So museum specimens are really important for documenting species in time and space and having records and physical specimens that we can look at. And so these are some examples of preps that we do, and these are all small mammals. One common prep is doing a skin. So this is a flying squirrel that's been skinned out and stuffed with cotton. It's really great for looking at the pelage or the fur color. We can also take samples of skin or hair if we need it. Usually accompanying these are skeletons. So this is actually the skeleton from this specimen. And so after skinning it, we gut the animal, we dry out the skeleton, and we put it in dermestid beetles, and those are actually flesh-eating beetles. And they sound really scary, but they're not. They only eat dried flesh, and so after sometimes even a couple days, the skeletons are pretty much devoid of all the tissues. We then put the skeletons in ethanol or into ammonia, and that takes out blood and a lot of the fats, and they come out looking like this. And the beetles do a really great job because all of these bones are still intact. Many of them are articulated, and so we can kind of see where they're placed. And another thing that I like to do on these is what's called half skins. So half the skeleton, or in the forepaw here, the feet are actually in with the skeleton, and the other half has the bones in the foot, in the skin. And so this can be really useful if you're studying foot morphology. You can actually x-ray these bones to see where they are in kind of space and relative to each other. If you want to take out the bones and measure them individually, this prep is really great. So this kind of hedges your bet of which one or what someone's going to do for a study. So this is a really common prep. Historically, a lot of people just did skulls. Postcranial, or bones that come after the skull, are really important for looking at morphology and locomotion. And so nowadays, most people also save pretty much the entire skeleton as well. Another really important thing that we take is tissue samples. So we take things like liver or kidney or heart, and we put that in a vial like this. And this is either preserved with ethanol or it's frozen. And this is great for doing genetic studies or histological studies or things like that. So these are kind of really common preps that are done, and then they're stored in museums, and they act something like books in a library. And so you might not know exactly what this will be used for, but someone in the future is doing a research project or a study, and they can go and access this book, if you will, and get the information they need from it. And so these might not be used for 100 years or 200 years from now, but at some point, hopefully somebody comes and gets really important information on these specimens.